Welcome to The Explainer. Today, we're cutting straight through all the noise and, let's be honest, the heavy confusion surrounding agricultural biotechnology. Look, few topics in farming generate this much passion or this much controversy. So our goal today is simple. We're going to give you a balanced, scientifically grounded breakdown of what these technologies actually are, and more importantly, what they mean for the future of your farm. Let's just dive right into that controversy, because this topic is incredibly polarized. On one hand, people hear the word biotech and immediately picture corporate monopolies and, environmental catastrophe, right? But on the other hand, proponents are out there championing it as the ultimate silver bullet solution to world hunger. So we're going to look at what these tools actually do, because the truth, well, it's far more nuanced and honestly far more interesting than either of those extremes. To start, we really need to ground ourselves in the facts. What exactly is agricultural biotechnology? If we move past all the sensationalized rhetoric, it's actually just a range of tools. It uses living biological systems, things like microorganisms or their derivatives, to develop or modify plants, animals, and microbes for agriculture. It's not magic, and it's definitely not inherently sinister. It's just applied science. Okay, here is our roadmap for this explainer. 1. The biotechnology spectrum. 2. Everyday farm biotechnology. 3. Accelerating crop breeding. 4. Genetic modification and CRISPR. 5. Farmer autonomy and seeds. And finally, 6. Navigating your farm's future. Section 1. The biotechnology spectrum. You see, biotechnology isn't just one single modern monolith. It's actually a vast continuum. We started way back in the 1900s with microbial biotech, then moved through TissueCulture in the 50s, adopted marker-assisted selection in the 80s, and only very recently arrived at the highly debated frontiers of genetic modification and CRISPR gene editing. Understanding exactly where a specific technology sits on this historical timeline is absolutely crucial to making informed decisions about it. Moving right along to Section 2, Everyday Farm Biotechnology. Let's talk about tissue culture, or micropropagation. This utilizes sterile labs to rapidly multiply exact, disease-free copies of plants. And you might not realize this, but almost every single commercial banana grown in India today is actually a tissue culture clone. It's the standard because they're totally virus-free and they fruit faster. The exact same goes for sugarcane, which avoids ratoon stunting disease this way, and potatoes, which get a massive yield boost from using disease-free mini-tubers. Now, this everyday microbial biotech has a massive economic impact for your wallet. Biofertilizers containing naturally occurring bacteria and fungi, stuff like rhizobium and mycorrhizal fungi, can safely reduce your synthetic fertilizer needs by an impressive 20 to 30%. Think about that. They literally fix nitrogen straight from the atmosphere right into your soil, making them an incredibly cost-effective and safe tool to use. Then we have biopesticides. These are derived from naturally occurring organisms and they control pests without all the risks of synthetic chemicals. Take Bacillus thuringiensis, or BT, for example. It produces proteins that are toxic to specific insect larvae. Or Trichoderma fungi, which suppress soil-borne pathogens. Far from being unnatural, these biological control agents are rapidly becoming the absolute backbone of integrated pest management across the country. Okay, Section 3, Accelerating crop breeding. Conventional plant breeding is, it's incredibly slow. Relying purely on visual selection to develop just a single new crop variety typically takes 10 to 15 long years. In a world dealing with rapidly changing climates and exploding populations, that timeline is just a massive bottleneck for us. And this is exactly where marker-assisted selection, or MAS, steps in as a massive accelerator. Instead of waiting years for a plant to grow just to see if it actually has a desired trait, breeders can now screen thousands of seedlings at the DNA level right out of the gate. In India, this technique successfully identified flood tolerance early on, resulting in Swarna sub-1 rice, which is a highly resilient variety that can actually survive 2 weeks of complete submergence while still maintaining high yields. Absolutely incredible. All right, let's dive into the deep end. Section 4, Genetic Modification and CRISPR. To clear up some major confusion here, creating a GM crop isn't some casual weekend lab experiment. It is a highly rigorous, heavily monitored gauntlet. It involves identifying a desirable target gene, isolating it in a lab, carefully inserting it into a plant's genome, and then conducting thousands of generations of testing in contained environments. All of that happens before any commercial approval is even considered. Looking impartially at the reality of GM crops in India, we have to note that BT cotton is our only commercially approved GM crop, and it's been grown universally since 2002. Now, it has undeniably reduced initial pesticide use and bollworm damage. However, the whole picture is definitely complicated by the development of secondary pests and pest resistance. Meanwhile, crops like BT brinjal and HT mustard have completed field trials, but they face ongoing regulatory delays amidst some very serious debates over environmental impact and corporate control. Now, there is a crucial difference between conventional GM and the new frontier of CRISPR. While GM actually inserts foreign DNA from a totally different organism, CRISPR acts more like molecular scissors, making precise edits to the organism's existing genes. Because it doesn't introduce any foreign DNA, it is currently being proposed for lower regulation in India, which could potentially speed up the development of highly customized, resilient crops. Section 5, Farmer Autonomy and Seeds. A major concern with advanced biotechnology is intellectual property. When seeds are locked behind corporate patents, it legally restricts farmers from saving and replanting them, which creates a really costly dependency. The alternative? Utilizing public sector research from institutions like ICAR and ICRISAT, who work to develop and provide improved varieties without those restrictive intellectual property conditions. Furthermore, open-source biotechnology and participatory plant breeding offer a really vital counterweight to that corporate concentration. By bringing farmers directly into the breeding process, having them evaluate varieties right on their own local fields, we ensure that the science actually serves the on-the-ground needs of local environments rather than just serving corporate bottom lines. Finally, Section 6, Navigating Your Farm's Future. Here is your ultimate farmer's checklist. Don't just accept or reject biotech based purely on ideology. Evaluate each technology on its own specific merits. Rely on peer-reviewed science from credible agricultural universities or ICAR. Make sure to engage with the policy processes that affect your livelihood. And above all else, always preserve your farm's traditional seed diversity for long-term resilience. If you're looking for a practical place to start today, here is an action step for you. The most accessible, lowest-risk entry point into agricultural biotech for most Indian farmers is simply incorporating biofertilizers and biopesticides into your current crop management program. The benefits regarding reduced input costs and significantly improved soil health are well established, and they are ready to be utilized right now. I'm going to leave you with this final thought. Agricultural biotechnology is neither the absolute salvation nor the sheer destruction of farming. It is simply a set of powerful tools. So the real question is, will you let ideology or will you let informed science guide the future of your farm? Thank you so much for joining me for this explainer. Keep learning, keep questioning, and I'll see you next time.